Yes, we're here again. This is question 4, basic agricultural genetics. And breed black-faced ram is crossed with a purebred white-faced ewe. The blue represents the ram's face color, which is dominant over the white face color of the ewe. Yeah. Then you can see graph, our graph here. The first real generation, the first generation of offspring, the parent bee, then this first real generation becomes the parent bee, and they get crossed, then they produce the second real generation, the generation of offspring. Right. So write down the genotype of parent bee. Okay. This can be a little bit tricky, but the ram is about even male. Okay, and this symbol may also tell you what it is or male. This symbol shows the sign for a male gender. Ewe is a female, which you can also see by this sign, represent female gender. So a purebred, they use the term purebred. A purebred means an animal with homozygous allele, an animal with homozygous or identical allele that we have purebred with. Okay. Homozygous allele or identical allele. So, capital letter B. Capital letter B represents what? Capital letter B means dominant. Means dominant. That means we have a dominant black color. Right. The sentence black ram is dominant over the white face color ewe. Okay. What does this sentence show us? Or what does it entail? This means that the black ram has two capital letters. Has two capital letters of B because it is homozygous. It has homozygous allele. Alright, and that Y E and not E has two small letters because it is a homozygous allele. And at this condition, we can say that the white E is homozygous recessive. Right. So yeah, so the parent B here we can say is BB, small letter BB. Yeah, that's the genotype of parent B. Indicate whether parent A and parent B are homozygous or heterozygous. As we have said, the term purebred, the terminology purebred, means an animal with homozygous alleles or identical alleles. And at this point, we know that okay, homozygous allele is the one that is applicable here because you don't have a heterozygous. A purebred is from two homozygous. And just homozygous. Oh, and 1.3, they want to support your friend. So we can say each parent has his or her own two identical alleles because they are pure breeds, meaning they have two identical alleles. Like two identical alleles. Each parent has his or her own two identical alleles because they are pure breeds. Meaning, we have two identical alleles. Okay. Open 1.4 identify the phenotype in the F2 generation is represented by F, G, and H. So to find this one, write your answer somewhere. Then I'm going to say I have a black one times a white E. This times that means I am replacing it with the white E. Then dominant BB has to recessive BB. Then my UCC, okay, my UCC takes place. Then I have my gametes. Then here you can see I have separated my gametes. Then this gametes, they cross with each other during fertilization. And the genotype for the first filial generation, I have the heterozygous condition, which is capital letter B and small letter b for all the offspring. For heterozygous offsprings, for heterozygous offsprings. Heterozygous just means I have the capital letter B and a small letter b on my alleles. Okay. Then the phenotype is all black. That's all black. Then I'm going to take two of my offsprings. Then now I make them the parents. Okay. I make them the parents so that I can cross them and get the second filial generation. My black ram, I'm going to cross it with my light blue. Okay. Then black ram and black E, that's my phenotype. Then heterozygous B. Okay. And heterozygous BB as my genotype. So I'm going to okay, this capital letter B and small letter b is crossed with the capital letter B and small letter b of the black one. Then this genotype here of this parent, right, it's going to go through meiosis. This parent is going to go through meiosis here, okay, and gametes are going to be produced. So I have B, capital letter B, and small letter b separated, then times capital letter B and small letter b, they are separated through the process of meiosis. Then comes fertilization. Then comes fertilization, and then I have my offspring, the second filial generation offspring. I have dominant alleles, homozygous dominant, which is BB, and I have heterozygous offsprings of BB, two heterozygous, okay, that are BB, and one homozygous recessive, BB, small letter B, two small letter b. So, right, I did it here, yeah? Then add to my question. They wanted F, G, and H. Then okay, as we can see, my answer says homozygous dominant BB. Okay, And you can see on the graph here we have BB. Right. That means sorry, F will be heterozygous condition, heterozygous offspring of BB, which is capital letter B and small letter b, choose capital letter B and small letter b. Right, that means F will be heterozygous BB, and B is going to be heterozygous BB, and also H is going to be our H one is going to be homozygous recessive because it has two small alleles.
Then we write F is for black color, G is for black color, H is for white colored offspring. That is our final answer. 4.1.5.
Phenotypic ratio and the phenotypic ratio in the second filial generation, the genotypic and the phenotypic. So if you have already done these workings on a scribble assignment, then you can be able to work out your phenotypic ratio speed. Then just write on paper, just say phenotypic ratio, then we say 1BB, which is capital letter B, then for dominant B's, then 1 dominant B's is two heterozygous B's is to 1 recessive, is to 1 homozygous recessive B's. Yeah, then the phenotypic ratio is going to be 3 black is to 1 white. Yeah, and right. Open 2. The table below shows the estimated breeding value, which is EBV for specific characteristics in Bosmara, cattle, and Boer goats. I think this represents your sharp. The table has species, characteristics, heritability. This is where Bosmara have Boer goat. Okay. Then characteristics: birth weight, first minimum weight, mid-tenderness, weight. Then Boer goats, birth weight, first minimum weight, face weight. Heritability, we have WWR, 88, 65, and so on. Then 4.2.1, identify one characteristic of each animal. Okay, and we have to identify the characteristics for Bosmara, one characteristic for Bosmara, one characteristic for the Boer goat. Okay, and this characteristic is going to be the characteristic that the farmers will select for breeding. Okay, for breeding, we have two animals, Bosmara and the Boer goat. Bosmara is the characteristic with high heritability. Okay, with high heritability than all of other characteristics. Going to do the characteristic that has high heritability than all other characteristics. Then for the Boer goat, we're going to choose one characteristic with high heritability on all characteristics. Then for Bosmara, the characteristic that has high heritability is what? Is mid-tenderness with 65. And for Boer goats, The one with high heritability is post-win rate with 60%. Yeah. 4.2.2 give a reason for the choice of characteristic. Why did we give this choice? The heritability of both characteristics is greater than 50%. Okay? The heritability of both characteristics is greater than 50%. As you can see, other ones have less than 50%. Okay? So it was more option for them to be chosen. They're less than 50 and don't qualify to be chosen. So you can use them this characteristic because heritability of both characteristics is greater than 50%. The highest heritability trait for Bosmara is 65, and the highest heritability trait for Dorothy is 60. Right? Open given breath. Give two reasons why Dorothy's farmers will probably not select the breath weight, loose weight, and lean weight. They can't choose them or consider them to be holding because they have a heritability of less than 50%. Okay. They have a heritability of less than 50%, and this will make the characteristics to be less controlled by the gene and be more influenced by the environment. And we don't want these characteristics to be influenced by the environment. We want these characteristics to be controlled more by the genes. But if we have this heritability of less than 50% or below 50%, this will make the characteristics to be less controlled by the genes and be more right. Right. Variation applies to the differences in characteristics between members of the same species. The differences may be caused by both external and internal factors. Name the environmental factor that has led to inject the following variations. 4.3.1 Animals at higher altitudes have darker pigmentation than those at lower altitude. Temperature. Okay, so temperature is the one that they are looking for because at high and low altitudes, so the temperature at high and low altitudes is not the same. The temperature at high altitude and the temperature at low altitude is not the same. Okay. Hence, exert temperature. Animals, okay, open that into animals are shorter than other animals with the same gene for tallness due to a nutrient deficiency. Animals are shorter than other animals with the same gene for tallness due to a nutrient deficiency. Okay, due to a nutrient deficiency. The correct answer is feed or feeding. Yeah, that's the one that we're looking for. And 4.3.3 goats kept on steeper slopes have longer and stronger limbs, and goats kept on shorter slopes. The correct answer for this is topography, or we can say relief. So basically relief describes the variation in height and thickness. That means the correct answer is relief, or we can say topography. Topography or relief. Because relief describes the variations in thickness of the earth ground, which matches with this and temperature. 3.4. Efforts kept in colder regions like thicker air and those found in warmer regions. So the efforts that are kept in colder regions like thicker air than those found in warmer regions. So this shows temperature variation. Temperature variation. Correct answer is temperature variation. And here, temperature variation. Cold temperature. 4.4. Assume that egg laying in Leghorn laying hen is controlled by 3 pairs of genes. The net production of a laying hen with the genotype EBGGKK is 60 eggs per week. Each edited allele contributes 5 eggs to the net production of eggs. 4.1 determine the net production of a laying hen with the genotype B, dominant B, recessive B, dominant G, recessive G, recessive KK. Right. We want what determine the net production of a laying hen with this. So this needs 6 dominant alleles. Okay, and 6 dominant alleles each adds a value to the overall base. Okay, so if here the base for the recessive genotype is 60 eggs per week, that means an addition of 6 dominant alleles will add more to that. So so that means a dominant allele will add 5. Okay, so add 5. Each one will add 5. So the dominant B will add 5 eggs and the dominant G will add 5 eggs. So dominant B, recessive B, dominant G, recessive G, and recessive KK is equals to 60 plus 5 plus 5, then it gives us 70. 4.2 name the genotype that will provide in 90. So for us to have 90, that means we have to genotype 2. All of the alleles in the genotype to be dominant, so that we can have 90 eggs. So that means we go for homozygous dominant B, homozygous dominant B, homozygous dominant K. And we have Now, 4.4.3 indicates the type of inheritance of control state. That indicate the type of inheritance that controls totally in left and the hands. That's polygenic inheritance. On 4.5 in animal selection, the following data infers is being collected for breeding purposes. The table has a live males, kg, and the number of animals. Okay. And we have these values, 13 values at the top, then 13 values at the bottom. Then it's open 4.5 and look at each minimum each other. We see a complete range of phenotypes of a quantitative characteristic. Okay. And these quantitative characteristics are showing weight gain range. Also. So this weight gain range of 134, 135, 136, 137, 138, 139, 140, 141. This is an example of continuous variation. Okay, because we are dealing with weight. Okay, this is continuous. Confuse this with other things because they don't want the name of the characteristic. They don't want the name. So, okay, they don't want the name. You can't say in weight or something. No, that's not what they are looking for. Here they're trying to just show you we are dealing with a range of phenotypes. Okay, every quantitative characteristic, and they're trying to show you a weight gain range of 134, 134, 135, 136, 137. This is live mass per kg, 134, 135, 136, 137, and all those things. This is continuous variation. And 4.5.2 determine the number of wafers. If 12% of the total number of wafers selected. Yeah. Right. So they want only 12% of the wafers that was selected. Only 12%. So to get started, so we need to get the total number of all wafers by adding the number of elements from each column. Okay. By adding the number of wafers from each column. So we're going to add 10, 15, 20, 30, 40, 60, 25, 65, 45, 45, 15, 10, 50. Then the total is going to give you what? 425. Okay. So we want 12% of that 425, which is the total number. Okay. So when you say 425, then you times it by 12%. Then we get the 1% answers, which is good. And 4.5.3, use the data to find the mass for an average wafer. Find the mass of an average wafer. So to find the mass of an average wafer, you need to get the total amount of values. Okay. So that means we will have to add those 134, 135, 132, 137, 138, 139, 140, until 146. Then the answer that you will get after adding all those values, you will get 1820. Okay, which is the total life mass. Okay, per kg. So you need to divide that total amount of life mass by the total number of columns. Okay, we have how many number of columns here? We have 30 number. So we're going to say 1820 divided by 30. So the answer that you get is 140. And that's 4.5. A normal commercial production unit. What will a farmer do? Okay, if I'm trying to put you in a condition where you are in a business, where you are in a business where you make production, where you make products, then you have to make decisions, okay, based on the knowledge about breeding and genetics. But keep that in mind. A heifers with the highest life mass. What will the farmer do with the heifers with the highest life mass? B. What will the farmer do with heifers with the lowest life mass? Okay, so a farmer, um, a farmer, um, what the farmer will do with heifers with the highest life mass is that the farmer will select those animals for breeding. Okay, it will select those animals for breeding. Okay, then what will a farmer do with heifers with the lowest life mass? Then the farmer will just slaughter those animals and sell those animals because he cannot use those animals for breeding. Right? So he will just have to slaughter them and sell them because they have no life mass. And 4.6, the illustration below shows a technique used by what? Farmers to genetically modify tomatoes. So you can see there's this tomato liquid and when this gene, that's the gene, and transferring the gene into the kidney of this tomato fruit. 6.1. Identify the technique about what is this technique. What is this technique? Its technique is micro injection. Micro injection. MICRO injection. Micro injection. 4.6.2 two advantages of the technique on question 4.6.1. So the advantage of the microinjection, the advantages of microinjection technique for the farmer. What is the advantage of using this microinjection technique for the farmer? So this microinjection technique, as we know, okay, what is this microinjection technique about? This technique is one of the techniques that are used in the genetic modification of plants and animals. Right? So, it is those techniques that are used in the genetic modification of plants and animals where the desired DNA is injected into the what? Into the nucleus of the recipient cell, right? So with that information in mind, we have to state two advantages of this micro-injection technique. So what will this technique do for the farmer, right? This technique increases production of crops and improves the characteristics of crops to have more quality, to be more nutritional and have more quality. Okay, so but because this set two, we have to separate them. Just so we increase production of crops and yeah, increase the production of crops, increase the yield of crops, and B, improve the characteristics of crops to have more quality, more nutrition. 6.3 suggests two socio-economic implications that plant produced from a micro-injection technique and other related techniques have for the farmer. Okay, so this is a tricky question. Yeah. So there is yeah, there are constraints here. Okay, they want only the social and economic effect. Okay, social will affect people socially, so okay, and economic effective affecting the farmer economically, so affecting the hectare, the money flow, or the cost of running the business and farm, some other cost, and all those things that economic effect. So economic effect is uh, both social and economic effects combined. Then they want to link this socio-economic implication to the farmer specifically. Okay, that means your answers have to be related to the farmer specifically. So, right now that we have that in mind, we can continue. So, A and yeah, A farmers cannot afford genetically modified crops. Okay, they cannot afford genetically modified crops. Okay, because they're expensive. They cannot afford them because these farmers are small scale farmers. Really, specifically small scale farmers, they cannot afford to buy genetically modified crops. It will be in it will be a heavy process for the small scale farmers to just make a revenue to be able to afford those genetically modified crops because they're expensive. So small scale farmers cannot always afford to buy those crops. Okay, because they're expensive. Then it is ethically questionable. Okay, as people think it is bad to intervene. Okay, it is ethically questionable as people think it is bad to interfere with their nature, and some customers will not buy from the farmer due to ethical concern. And yeah, so the part that we get to the other part, and some customers will not buy from the farmer due to ethical concern. This is social effects. What social effect? People. What people say. Customers say that the people. Okay, they are affected by this social effect. These people say we will not buy from the farmer because of what the farmer does. The farmer buys GM crops. So because these GM crops are ethically questionable, as people feel it is fair to interfere with their nature. So some consumers will not buy from the farm due to ethical concern. That's why, and that's it.